Hello guys, so a quick one. In this video, I will be reviewing the parts of the microscope. But firstly, the microscope is an equipment used in the laboratory to magnify or view objects that are very tiny and cannot be seen with the naked eyes. Okay, so I'll go into the review now. So this is a light microscope. This is a light microscope, and I'm going to be going into the parts of the light microscope. Basically, the microscope is divided into three major parts: the head, which is this part, the neck or the middle, and then the base. The base. Okay, now this part is known as the ocular lens. The ocular lens. Since they are two in number, you can call them binocular lens. Binocular lenses. Now it has a magnification of times 10. This magnification is a constant magnification. It doesn't change, so it has a magnification of times 10. Moving downwards, this is what we know as the revolving nose piece, and the revolving nose piece helps to attach the objective lenses. Now these lenses are called the objective lenses, and they are basically 4 in number in a light microscope. Now starting with this is what we know as a times 4 objective lens. It is usually marked with a red ribbon. Times 4 objective lens. This is the times 10 objective lens, usually with a yellow ribbon. This is the times 40 objective lens, usually with a green, lilac, or any other color ribbon. Now this is what we know as the oil immersion lens, which is times 100. Times 100 oil immersion lens. For you to use this lens, you need an immersion oil. Okay, so these three lenses, the times 4, the times 10, and the times 40, are called wet prep lenses. They are called wet prep lenses because they are used to view wet preparations. Wet preparations. Meanwhile, the times 10 is used to view dry preparations like gram stain slide and malaria parasite slide, and also to count white blood cells. Okay, so that's that. So the next, this is the mechanical stage. Now the mechanical stage helps to hold the slide. You place the slide on the mechanical stage, and this is the slide here. Okay, now it is placed on the mechanical stage. You also have the clips attached to the mechanical stage. The clips helps to hold the slide in place. Okay, so that is that. Now moving forward, this is known as the coarse adjustment. The coarse adjustment helps to focus your organism you are trying to view. Now it helps to move the mechanical stage upwards and downwards, like you can see here. Okay, so the next knob here, the smaller knob, is known as the fine adjustment. Fine adjustment. Now when you have focused with the coarse adjustment, the fine adjustment is used to make sure the organism in your slide is clear enough to be seen or to be examined. Okay, it helps to increase the contrast of your organism, right? So moving forward, if you observe below the microscope, you will notice that there is something here that is called the condenser. If you observe, you will notice that there is a condenser under the microscope. And this condenser, what's the function? It helps to allow light to pass through the light source to the organism. Okay, now the condenser is like a vacuum or a cylinder that helps to gather light and make it hit the organism on the slide. Okay, so attached to the condenser is what we know as the iris, or you can call it the diaphragm. Now this diaphragm helps to control the amount of light that passes through the condenser. Okay, so that is that about the condenser. Now over here is the light source. The light source. This is the light source. So basically, when you put on the microscope, you can see the light has come on. So that light hits the organism on the slide, and you are able to view it on the ocular lens. Okay, so these are the basic parts of the microscope. Okay, Now to the principles of the microscope. Basically, when you put an organism on a slide and you put on the microscope, the light passes from the light source through the condenser and hits the organism, and you are able to see it on the ocular lens. Okay, now the ocular lens and the objective lens are closely associated. Yes, because the both lens gives you the magnification of your specimen, the specimen you are viewing. Now, the magnification of your specimen is the product of the ocular lens and the objective lens. So, if the ocular lens is times 10 and it's constant, let's assume that we are viewing with times 40. So, since it's the product, it will be 40 times 10, which will give us 400 magnification. So, if you are viewing, whatever you are viewing under times 40 objective lens will be 400 magnification. The same thing happens with times 10. Whatever we are viewing with times 10 objective lens will be 100 magnification, and so on. All right, guys. So in my next video, I'll be teaching you how to use the microscope, how to focus the microscope, and we are also going to try to focus some specimen and see maybe some microbes under the microscope. Okay, so see you in my next video, and don't forget to like and share. Thank you, and see you in my next video.